had welcome everybody. I hope you all had a great summer break and you could take a bit of time off and without too much heat. So we are very happy to resume the animal genomics monthly meetings, and this time we have a very interesting speaker as usual, Brenda Murdoch, who will tell us about sheep pangenomics, and Francesca will soon introduce her. I'm very happy to resume this season with Francesca Bernini and Jose Espinosa, who have been supporting the animal genomics group tirelessly over the last years. It's quite interesting that we are now starting this group for the third year. So I think we've been running in total about nearly a bit less than 2 years in total, and this one will be a third year. So it is something going on nicely and strong, and we can see it with the attendance that is increasing every time. So thank you all for being here. I know that it's not easy to find a good time that is suitable for both the US, Europe, and now we also have requests from New Zealand and Australia. So we have to try to figure out something at some point. Let me remind all of you that if there is any topic that you'd like to talk about in this channel, please reach out to Francesca or Jose, and we are always very happy to have new groups. Right now, I think we have speakers till early 2026, but there are plenty of slots to come for the next year. So if you have, if any of you have any interest in talking on this channel, please reach out to us. And without any further ado, I will leave the floor to Francesca, who is the one responsible for inviting Brenda. So thank you, Francesca, and we'll therefore have the honor of making a brief introduction of Brenda. Thank you, Francesca.
Thank you, Cedric, and thank you everyone for being here. So we are starting strong since for today's talk, as Cedric mentioned, we are delighted to welcome Dr. Brenda Murdoch and Temitayo Olangunju. I hope I pronounce it right. I'm sorry if not. Okay, thanks. So Brenda is a leading researcher and a professor in livestock genomics at the University of Idaho, with a particular focus on structural variation and pangenome. And she is currently keeping the sheep pangenome project. So her work has greatly advanced our understanding of genetic diversity in sheep, but also other livestock species. And with her, Temitayo, he's a postdoc at her lab, who has been working hands-on on the sheep pangenome project. And together they will share their insights and experiences in building and analyzing the sheep pangenome. So please join me in giving them a warm welcome. And Brenda and Temitayo, the stage is yours.
Okay, thank you very much. We are just going to pull up our slide set here. Okay, hopefully everybody can see. Okay, we're good to go. Okay, well, first of all, I really just want to thank everybody for the opportunity to share the work that we've been doing here regarding the sheep pangenome. And as mentioned already, my name is Brenda Murdoch, and I have with me Temitayo, and we are just so pleased to share what we've been doing. Maybe. So I really just wanted to start off by acknowledging that the work that we've been doing in the sheep pangenome is actually a collaborative effort. So it's not just Temitayo and myself, even though we have the pleasure of representing it. And so acknowledging both the USDA funding for allowing us and providing financial support for us to do this, but additionally, all of the other collaborators that have contributed to this particular project. So just to kick things off, a high-quality reference genome is an important resource for all genomic and genetic research. And I think everybody probably on this call really knows that and appreciates that. And I just wanted to sort of start off by sharing that many of the people that have contributed to reference genomes in the livestock region or field are not really genomic assemblers by training. This is something that many of us have embarked on, mostly because we really understand and need this resource to do all of the research that we really wanted to do. So one of the first genomes that I've contributed and helped assemble was the bovine pangenome. Many years ago, we needed that pangenome in order to really understand and develop the SNP reference panel that we were doing. And so again, you know, this is what we've been doing is because we need this resource, not because we're genome assemblers by training, but because of the importance of this resource, many of us have taken on this work. So until recently, and I'll just say complete genomes were very difficult to assemble. So as technologies have changed, our ability to create better reference genomes has improved. But most of the reference genomes that you see on NCBI or Ensemble would not be considered complete in the sense that they're not telomere to telomere assemblies. But more recently, we have been able to assemble telomere to telomere assemblies, but they are still a little bit difficult for some species, and actually sheep are one of those species. And so if we're trying to get all the way from the telomere through that very gene-rich region to the other telomere, this is now possible. And I say that until recently they weren't difficult, but some species are still challenging to do this, and sheep is one of them. But with newer technologies, long read technologies in particular, we can now actually assemble these genomes from telomere to telomere. But despite the completeness of a reference genome assembly, it is still a single representative of a species. So it does not adequately describe all of the genomic variation across the population within those species. So we've worked for years and years to get these high quality reference genomes. And now that we have complete T to T assemblies, we do appreciate the fact that that is only one individual. And these pangenomes are something that will allow us to capture more of the genomic variation within that species or within a given population. And so when we think about some of these species, in particular sheep, they are very rich in biodiversity. And what I'm showing you here on the left is a phylogenetic tree, and just kind of looking at when sheep diverged from the caprinae was approximately 5 million years ago, depending on which study. So there's a little bit of variation in there. But and so just trying to understand how these species have evolved and how they have diverged. And that's part of what we're trying to do in this pangenome. Now, if you look at the panel on the right, here we're showing you a publication from Daly that comes out of, that just came out in 2025. And they're talking about what they did is looked at a number of sort of ancient DNA from sheep and trying to trace back the origin of Ovis aries. And what they show is that Ovis aries does in fact evolve and diverge and from the mouflon. And really, if I can just point your attention to some of these maps, and you can look at the movement of sheep as they have evolved over time. And as they evolved over time and moved into these different regions, they've exhibited different phenotypes, and these phenotypes have allowed them to adapt and are different for different regions that they've moved to. So in reference to the sheep pangenome project, These are just some representative pictures of some of the different phenotypes that we were looking at. And I want to just sort of pause and point out that the approach that we took for the Sheep Pangenome Project is a little bit different than some of the other pangenome projects. And what we did is we really targeted specific breeds that have evolved to have different phenotypes of interest. And so just for example here, Merino, the first breed that the first picture are sheep that have very good wool. And so if you go and buy a wool suit or some of the wool fiber, they usually use breeds that, such as the Merino, that has a very, very nice wool coat, as opposed to some of the other sheep. The sheep that's next to it here, this Awasi sheep, this sheep is evolved to do very well in semi-arid environments. So as we consider climate change and warmer Earth, we really need to understand those breeds that have adapted well in these types of environments. Now I'm just going to go right down from the Awasi to this Suffolk breed. This black-face breed is a breed that is very rapidly growing and is a very good meat product. But this breed of sheep is actually very susceptible to parasites, as opposed to this picture over here that I'm showing you, the St. Croix breed. The St. Croix breed have evolved in Mediterranean climates and have adapted to do very well and are resistant to parasite infections. So trying to understand the difference between the Suffolk and the St. Croix and how one breed is adapted to parasite resistance versus another is one of the important aspects of this study. And then I can just, there's a number of breeds here, and just bring to light the fact that some of these breeds are in bold. The breeds and the sheep that are, whose names are bolded, are the ones that we created haploid genome assemblies for in order to advance this pangenome. And then you might see a couple other sheep that are not bolded. And so what we did is, in addition to the new genome assemblies that we added, we took a number of high quality genome assemblies that were in the public domain, so that we could really have a very complete set of representative genomes and their phenotypes. And then I'll just bring your attention to the very end. Those are the breeds that are in blue. And again, this is the Mouflon. So what we think is the more ancient or the more historic breed of species, actually different species of sheep that these domestic sheep evolved from, as well as the Argali right beneath it. And then finally, the bighorn, Ovis canadensis. And I really just wanted to take a couple minutes here. So this is one of the species that we did a T-to-T assembly for, and that is now available on NCBI, and hopefully our paper will be available for people to read very soon. But one of the reasons why we included this particular subspecies of sheep is that they are very susceptible to bacterial infection and often succumb to a bacteria called Mycoplasma ovonemoniae. And it turns out that many of the domestic sheep, if they're exposed to Mycoplasma ovonemoniae, they will get sick, but they will recover. They'll have a runny nose, and they will have some traits and some symptoms of having this disease. Whereas these bighorn sheep, it is mortal for most of them. So lots of times they will actually die. And so in the Pacific Northwest, where we have a bunch of public land where we may have domestic sheep and bighorn sheep that might intermingle, this is a very important issue for producers and ranchers out here, as well as an important issue to try and understand why these bighorn sheep are so susceptible to this Mycoplasma ovonemoniae. And so with that, I hope I've really sort of illustrated why pangenomes are important. And I am actually going to turn over how we did this pangenome and a little bit more of the specifics to Temitayo. So here's a nice picture of Temitayo receiving an award for this work at the EGBT conference.
Thank you so much, Brenda, for the introduction and for laying the foundation for my talk this morning. So continuing from where Brenda stopped, the importance of a reference genome cannot be overemphasized. However, that single reference genome is not sufficient, it's inadequate to capture all the variation that we have between different breeds of sheep and between populations of sheep. So what are we talking about? We have what we refer to as reference bias, where you have, for instance, we have this representative single reference, where you have these supposed gaps, L1 and L2, in the reference relative to perhaps two other breeds. And for these other breeds, if you have variants, whether single nucleotide variants, structural variants, inversions, or copy number variants, within those other breeds, for instance, this yellow region for this breed in green, or this lime-colored regions in this maroon-colored breed, you see that these regions are not present in that reference. And because of that, any attempt to try to characterize the variants that we have in these two breeds, the green and the red breed, using that single reference will not be able to show up. They will not be captured by that single reference just because the single reference does not have those regions. And how do we treat this? Of course, based on the pipeline that is used for variant calling, aligning short reads first to a reference, because of the fact that those reads are not present in the reference and will not align to the reference, what we do is to drop those reads while filtering the alignment file, and eventually those are going to be missed completely. So this reference bias is one of the problems that using a pangenome would help us to solve, because you have as many variants as possible represented in the pangenome, such that aligning short reads, you have those reads aligned to the proper location where they should be, because those variants are actually captured in the pangenome. In addition to that, we expect to be able to better characterize structural variants in these breeds. And for instance, Homia, which is the online Mendelian inheritance in animals, a website that catalogs known variants in animal breeds, has this statistic to show the proportion of SNPs, of small indels and structural variants that we have for the animals that are represented here. And looking at this proportion, we'll see that the structural variants and small indels, they have smaller proportion compared to the SNPs. And I must also say that in Omia, the structural variants are categorized as variants that have at least 20 base pairs, which is a little lower than the widely accepted 50 base pair characterization for SVs. What I'm saying in essence is that a number of these that are characterized as structural variants would actually even fall within the small indel category. And looking specifically at sheep, we'll see that we have a very low proportion of the variants as structural variants compared to the rest of the types of variants. And this low proportion is not, is caused mainly because of the fact that most of the tools that are used for variant calling, they don't do well when it comes to structural variants. Of course, they do well when it comes to calling SNPs, etc. And from this, we have very beautiful work that was done by Kosuji et al. They did a very in-depth analysis to show the shortcoming of these variants, and also by Zook from the Human Genome in a Bottle Consortium. So this main challenge that we face with characterizing structural variants is one of the challenges that we also hope to be able to solve with using a pangenome. Now, the pangenome is able to carry this out using what we call a variation graph. And with the variation graph, you have multiple genomes of multiple individuals. It could be between species, as we have in sheep pangenome. And then these are represented as a single graph where the regions that have common sequence are represented as a single node, like I have in this illustration. And then the parts of the genomes of the individuals that make up the pangenome that are different from others, you have them represented as smaller nodes, such that to recover a particular genome is just a single walk through the path that is traversed by each of the nodes in the genome graph. And then the nodes actually are segments of the genome, which could range from one base pair to as long as the node segments could be. It's also important to point out that this variation graph becomes very easy to handle downstream, because this variation we have in the different genomes that make up the pangenome are encoded in a VCF format. So it becomes easy to work with downstream. Now, specifically for this pangenome, we're using a total of 23 domestic breeds and then 3 subspecies, as we have listed here. And these breeds that we have in purple, as Brenda has mentioned earlier, these were the breeds that we assembled ab initio, that was generated and assembled ab initio. These breeds in blue were the ones that we sourced from public repositories, mainly NCBI. And Rambouillet here, the last on this list, is the current domestic sheep reference. So we added it to the pangenome as a means of also referring to previous studies that have been based on the Rambouillet. Then in addition to that, we talk about these other 3 subspecies, which is the bighorn, which we also generated ab initio, then the mouflon and the agali. So I must also say that the approach to produce these assemblies for the ones that were ab initio generated was crossbreeding these animals, such that would be able to separate the F1 into the component assemblies of the sire and the dam. And going into these assemblies are hi-fi reads and ONT ultra long reads. And in some cases where we had Illumina data for trio, we used the trio assembly approach, which helped to properly face and separate the haplotypes of the parents from the F1. So looking at the assembly statistics of the assemblies that went into the pangenome, we would easily see that these assemblies are high quality, especially the de novo assemblies, going by the NG statistics of these assemblies. And this is also important because a quality pangenome would require to have quality assemblies going into it. And I'll point to this further down during this presentation, as we'll see. And our assembly lengths varied between 2.7 gigabases to 3 gigabases, where we had config N50 of between 78 and 111 for the assemblies that were used for this pangenome. So talking about the actual building of the pangenome, we used 2 of the main approaches that are being used, especially for the animal community. And for this, we used PGGB and Minigraph-Cactus. And I just want to point out the salient differences between the 2. And PGGB is known as a reference agnostic pangenome builder because it does an all-to-all alignment of all the genomes that we have in the assembly. So it is not the case that you have a dedicated reference assembly at the point of building the pangenome. On the other hand, Minigraph-Cactus requires you to select a single genome as the reference, as the backbone for the cactus alignment. And then from the backbone, it progressively aligns each of the other assemblies to the backbone, only adding those parts that are different from the reference. And because of these two different approaches, of course, there are slight differences, or there are differences in the outcome that you have from the pangenomes that are built. So that is why we decided to use the two approaches to be able to compare and see how the different tools capture the variation that we have in the different breeds that make up the assembly. So the steps that were used for building the pangenome specifically, we start with, excuse me. Thank you. So for the steps that were used, we start with bringing together single haplotype assemblies. Remember, from the crossbred animals, we have single haplotypes representing each breed. And then for the public assemblies, we needed to prepare the public assemblies because these assemblies have a run of hens that were used to scaffold the chromosomes. And this does not do well. When you have runs of hens, they don't do well when you're doing alignments and all. So preparing this, and also to identify the best of the assemblies to be used as the reference. Remember, for Minigraph-Cactus, it requires a reference. So we had to select the best of all the assemblies, the most contiguous, which in this pangenome was the Frisian assembly as the reference. So going from there, we passed the assemblies to the different pipelines. And for the PGGB pipeline, you could actually pass your assemblies in the PGGB pipeline, and then it runs all the way to generating your VCF files for you. But in this particular case, we couldn't run it all the way, and that was because of the naming of the chromosomes. And for PGGB, they recommend using the pan-SN naming convention, where you have the sample, which in this case will be the breed name, and then the haplotype. Then you specify the chromosome. As you will see for this illustration here, the separator for the sample, the haplotype, and the chromosome in this case is the hash character. But when you use this approach for when you have multiple contigs for a particular chromosome, it becomes challenging to be able to specifically identify each of those contigs. So it's not possible to have, for instance, having multiple chromosome 1 representing maybe 3 contigs for the whole of chromosome 1. There has to be a way to separate each of those contigs. For instance, you could use chromosome 1A, chromosome 1B, chromosome 1C. But I say this because this was the initial approach that was used, but we later found out at the time of trying to index that this was not amenable to VG indexing. So we had to change to an approach that is a naming approach that was amenable to VG indexing downstream. So that's why adopting this new approach where you have the sample name, the haplotype, and then the chromosome. In addition to that, you separate each of the contigs within a chromosome with separate numbers, which is the face that we have in here. So with this new naming approach, PGGB does not like this naming, and then it wouldn't work with this to generate the final VCF file that you need to represent the variation that you have in the pangenome. So it has to be done manually using VGDeconstruct, and then smoothing with VCF Bob and VCF Wave. So from the PGGB pipeline, in addition to getting the SB characterization, you also could have the GFA files representing the graph from the different breeds that went into the pangenome that could be used as a reference in quotes now for other downstream analysis, which one of the first things you want to do for such downstream analysis is to index the pangenome graph. So for the Minigraph-Cactus pipeline, there was no problem with respect to using this naming approach. So one is able to go directly from the pipeline to just filtering the graph and then indexing. Then from there, you could directly get the characterization of the SVs that you have in the graph. And so regarding utility of the pangenome, three main approaches that the pangenome could be used, which is to use a personalized pangenome, where you get to identify just a subset of the breeds that went into the whole pangenome construction, and using only that subset for analysis. Then another thing could be to go through the pipeline of reads alignment and genotyping, which is mainly done, in most cases, is done with VG giraffe. And then the third option is to carry out a direct comparative analysis between the different breeds that went into the pangenome to see how they are related at specific loci of interest. So for this pangenome, we decided to build this pangenome in two different sets. So first and foremost, based on the fact that we have the domestic breeds on one hand, and then the subspecies on another hand, we decided to build a pangenome for the domestic breeds on one hand, and then another one for all the domestic breeds, including the subspecies on another hand, just to see how much variation the subspecies actually contribute to what we have in the domestic breeds. So since we're using two tools to build this pangenome, we eventually had to build two for each of the two sets, such that eventually we had 4 different pangenomes built from a combination of the set 1 of the breeds, which make up all the domestic breeds, and then the set 2, which was all the domestic breeds and the 3 subspecies that were used in this pangenome. So looking at the final product of the characterization of the variants, not variants now, looking at the nodes and the hedges that we have in the pangenome, we'll see first the set 1 PGGB pangenome, which is the PGGB pangenome of all the domestic breeds. We see that based on the number of nodes and the hedges that we have, calculating the complexity of the graph using the density of the graph as a measure, we see that we have a higher complexity of the graph compared to what we have for the Minigraph-Cactus set 1 graph from 2.9 to 2.1. And we also see the same pattern with the set 2, which is all the domestic breeds and the 3 subspecies between PGGB and the Minigraph-Cactus, where you have the density for the set 2 PGGB was also higher than the set 2 Minigraph-Cactus. And what this tells us is just to reiterate the fact that based on the approach that these two graphs are using to construct the pangenome, you have different levels or different complexities or different representations of variation in the graphs that imposes that level of complexity on the graph that you have from the pangenome. And comparing this with some other pangenomes that have been published, for instance, looking at the Human Pangenome Reference Consortium version 1 pangenome, we have similar statistics to what we had for PGGB, although they had more haplotypes. They used 90 haplotypes compared to the 23 that we use for the PGGB pangenome. And for the African apes and the orangutan, we see that for the orangutan, we had a similar density, although the other statistics on nodes and edges were a little different. And this iterates or also helps us to see that we have a similar result, which is comparable to what has been done in some other mammals. So looking at the statistics of the SNVs that we have characterized from the pangenome, these have been separated on the left panel to only SNVs, which are SNVs that have a length of at least 50 base pair. and then we have the short indels. So we have to separate it because the numbers of the indels were, of course, drafting out the representation of the SNVs in the graph. And we see clearly, which is what is expected, that the SNVs that we have in the set 1 of Minigraph-Cactus compared to set 2 were lower. And then similar thing was observed for the PGGB graphs, also in terms of the indels that we have on the right side of the panel. But something important to just point out from looking at this graph is the increase in the number of SNVs that we have with the introduction of the 3 subspecies to the 23 domestic breeds that we already have. And we'll see a difference, an increase of between 22% and 48% of the variation that we already have in the domestic sheep alone pangenome, which is the set 1, compared to the set 2, which you can see from here. So from the Minigraph-Cactus, the insertion SNVs, we see an increase of about 47% of the number of variants captured. Then for the deletion SNVs, about 48%. Similarly, for the PGGB SNV, we see an increase of about 35% for the insertion SNV compared to 42% for the deletion SNV. And then the same pattern was also observed for the indels, where there was an increase in the variation that we have when the subspecies were added to all the domestic sheep. So this also further iterates or underscores the fact that the more the number of breeds that you have to your pangenome, then the higher the number of variation that you are able to capture from those other, from the new breeds that you're adding to the pangenome. So validation of the pangenome. Because of the fact that you have so many assemblies going into the pangenome, there is, at the moment, not a single approach to validating that the pangenome that you have correctly represents the variation that are supposed to be in these breeds. And there are so many different ways to actually do that now. And one approach that we are using is at the point of specifically analyzing the pangenome for some traits of interest. So looking at traits of interest between all these breeds, when we have the catalog of variation that we extract from the pangenome, then what we do is to look further at the alignment, the sequence alignment of the reads that produced the particular graph, the particular genome, to see the alignment of the reads, the concordance of the reads to the genome that produced the graph. And what we see in this illustration is 2 examples of such, where we are looking at this deletion variation on chromosome 2, which is falling on this particular gene PCSK5, and another chromosome 9, which is falling on ANGPT1. And what we saw from the pangenome made us to go back to the assembly to check whether what we are seeing represented in the pangenome truly represents the variation that we see between these breeds. And so it was discovered that alignment of the high-fi reads that produce the assembly back to the assembly, we could see that we have this discrepancy at that particular breakpoint of the variation of interest. We see both, for these examples I'm showing, we could see this discrepancy between the reads and the assembly at that particular locus. So this, of course, made us to conclude that what we are seeing in the pangenome at this particular locus is not real. And of course, such information is not to be processed further. So one thing I'd like to also point out from looking at this particular reads, which we also noticed on some of these other instances, is these kinds of events occur where you have a homopolymer run or a run of dinucleotides. In this case, we have a run of Ts. And then in this other one, we have the dinucleotide TG. And these are likely assembly artifacts introduced by the assembler And then a way to probably mitigate this would be to perhaps go deeper into doing some manual curation and polishing of the assembly that is used in building the pangenome. So talking about some of the challenges that we encountered in the process of building this pangenome, like I mentioned earlier, config and chromosome naming initially was challenging until it was revealed. Of course, the recommendation when using PGGB is to use the pan-SN naming. So that's we had done that until we realized much later downstream that that was causing problems for the step of indexing the graph. So that had to be modified, had to go back and reconstruct in a manner that is amenable to indexing with VG. And what that did initially was to blow up the computation time such that, I mean, we ran some of these graphs for up to 3 weeks without getting a successful indexing until it just had to be terminated because of that wrong naming. And eventually, after the naming was corrected, it was more amenable and then the indexing was successful. Then the other thing, which is why I mentioned the importance of removing the hens that were used to scaffold the contigs of the chromosomes from the publicly accessible genomes that were used, is the run of ends also increase the computational requirements. It blows up alignment time, and of course, it causes problem downstream for using the variation graph that is produced. So that is the reason we had to make sure that all the hens, because normally for submitting to such public repositories, you're expected to scaffold your chromosomes with a run of ends. So using that, we're able to eliminate this challenge and make sure that the pangenomes are amenable to use downstream. So another challenge I just would like to point out is the direct utilization of the pangenome graph with short reads alignment. So aside the challenge of the long runtime of indexing the graph in preparation for short reads alignment, it was also difficult to be able to successfully align a whole genome sequence short reads data. For instance, in this particular test, since it was difficult to align a complete sample, what we did was to just subset to just have an idea of how long it was going to take to really carry out this alignment. So for just 2 million paired end reads, took about 2 hours, 5 minutes. That's 2 million of the subset of 291 million reads. And the memory per CPU was set as about 4 gigabytes, CPUs per task, 36. Then I'm pointing out this number of tasks because it was possible to specify more cores to have more parallelization, but unfortunately, VG-Giraffe was not amenable to using multiple processors. It was only limited to a single processor. So with this, it was difficult to be able to use more computational resource and to be able to speed up the requirement. So extrapolating from this data, it was going to take about 6 days of work time to align a complete sample, a single WGS sample to the pangenome. So I'd mentioned earlier about the 3 main approaches of utilizing the pangenome: personalized pangenome, reads alignment, and genotyping, or direct comparative genomics. So what I'm going to show in terms of utility of the pangenome is the comparative genomics approach, where we looked at a locus that is well known in sheep that is responsible for on phenotype, whether the sheep is going to be horned or polled, and that is on the RXFP2 gene. So we looked at this particular locus on the pangenome, and that locus is caused by about 1.8 kilobytes insertion in the 3' UTR of that RXFP2. And looking at this, that locus was identified at this point where you have this red arrow pointing at. And we were able to go back and confirm the presence of this on some of the some of the animals that we know that carry this phenotype by aligning short reads from individuals that have the phenotype to a subset of the assembly. So what we have here, we have a set of horned merino and polled merino, then horned dorset and polled dorset, which actually confirms from the locus of the alignment, which confirms that the presence of this, what's it called? The presence of this insertion was attributed to the polled phenotype that we have. So in a nutshell, we've presented a sheep pangenome of 26 breeds, which were based on a careful selection of diverse traits of sheep from different parts of the world, in addition to some that were sourced publicly. And we showed that this pangenome was built using two state-of-the-art approaches, PGGB and Minigraph-Cactus. And from there, showed that the computational resources requirements for the pangenome for genotyping, short reads especially, is high, especially for the pangenome graphs that are built with PGGB. And finally, we'd just like to say that the use of pangenomes actually holds so much promise. And I know that's being a very dynamic and rapidly evolving field. There are so many improvements that are currently being done, and then there are still some that are still required to be able to make this whole tool to make it amenable for use for further computational, for further genomic and genetic utilization. So at this point, I would like to hand over briefly to Brenda for the last slide.
Okay, and I just wanted to, I was requested to give a quick shout out to PEG 2026. I see we have a number of questions and we're happy to take that. And just to remind people that are outside of the US, if you want to come to PEG, please just apply for the travel grants. The organizers are looking to increase the number of international participants in this particular conference, and we would be happy to see some applications and award some travel grants from our international partners. And with that, we're happy to take any questions, and thank you all for your attention. Maybe we'll just stop sharing here.
Thank you so much, Brenda and Temitayo. It was a great talk. I see a lot of questions in the chat. So if the people that have a question want to unmute themselves, you can either say hi and ask the question, or I will read it.
Okay, we can start. I think the first question that I am seeing, oh no, I lied. Go ahead. Just a reminder that the talk is being recorded.
Maybe I can help here. I've been following. So Chris was the first one to have a question. So Chris Tuggle, do you want to unmute yourself and ask your question?
Sure, I can do it. Thanks so much, Brenda and Temitayo. I learned a lot about pangenomes. But I had a question. So you described the difference in results between the two softwares. And can you explain the density calculation? Because as far as I understand, networks that have more nodes and more edges, might be giving you more information. So I was just wondering why you're focusing on density.
Okay, thank you so much for that great question. So the calculation for the density, since it's a non-directional graph, is a function of 2 multiplied by the number of edges divided by the square of the number of nodes. So that is the estimate for calculating the density in the graph.
But Chris, to your point, that's correct, right? So the more complex and the higher the density of that particular graph. It gives you a little bit more information, but the cost on the other side, when you're trying to maybe map a whole genome, sequence back to it costs a little bit. So, you know, we do these pangenomes in hopes to capture as much diversity as we can, but you can see when we add in those other subspecies, we are capturing a lot more diversity, and that makes that graph way more complex. And so, you know, just trying to think about ways to answer and ask questions on maybe, I guess I could think of it as a macroscopic scale and also microscopic scale.
Okay, well, so which, is there a preference, or do they each provide information that's important? So, I mean, do you need to always run both, or?
Yeah, I don't think you absolutely need to run both. I think if you run Minigraph-Cactus, you'll be a little bit quicker, but you will potentially miss some of that complexity. Whereas if you run the PGGB, you will capture more of that and retain more of that complexity, but your compute time will cost you a little bit more. And again, the more variation in the samples that you have going into that graph will also contribute to that.
Okay, thank you.
Sorry, can I just bounce in here and ask a clarification? So because Mini-Cactus is progressive, is there no possibility that some variations are counted several times? And that would explain the larger number of edges you get sometimes. So don't you get some kind of potential duplications?
So I think in the pipeline, this has been taken care of because eventually you have filtration steps, etc. And then eventually you wouldn't see such duplication that you're talking about in the final VCF that represents the variation that you have in your graph. So I think this is being handled internally such that you don't have double counting of the variation.
I don't mean exact duplications, but I mean slight variations that will naturally be represented as a variation inside a variation, but may end up as two separate structural variation, if you see what I mean. So if you were making the graph yourself by hand, you would have a structural variation and a small variation inside the variation. But because of the way things are computed, is there a chance that you occasionally end up with two different structural variations?
Okay.
That are composite of one another.
Okay, the opposite of one another. Composite of one another. Yes. Yes. So what happens with Minigraph-Cactus is when you have these kinds of composite, like you mentioned, they are decomposed to represent individual variation. So it's one of the reasons, or one of the ways where this is different from PGGB, because PGGB represents nested. It gives you nested. It could give you nested loci that has nested variation. But Minigraph-Cactus tries to linearize such that you don't have nested. So at a particular locus, you could have multiple or have different types. But then eventually, when it gets represented, you have them as individual variation.
And does this explain the question of Chris that you end up having more edges and more, you end up having more nodes, but the density of the graph is a little bit lower because you have less edges going into every node. Is this correct?
Yes, it has to directly. Because of the fact that in PGGB you could have it nested, which captures as many of the variation as possible. So you have a nested such that you have multiple edges going to a particular node, where in Minigraph-Cactus you have it decomposed.
And I have one last tiny one, and I won't talk again, but we have an NF-core pipeline for Pangenomes. Have you had any chance to look at it, or do you know does this ring any bell? Because this channel is actually about exploring the use of NF-core resources, and so I don't know if you guys are yet familiar with NF-core or not, but the bottom line is that these are community developed pipelines, and the most significant improvement over the last years have been the introduction of modules, where pipelines are composed of modules, and groups can contribute either through pipelines or that can be collectively maintained, or through modules that can be incorporated in pipelines. And for instance, important projects like the Darwin Tree of Life are not actually part of NF-Core. Their pipeline are not in NF-Core, but their pipeline are made of modules that are in NF-Core. And so when they make modules, they deposit them into NF-Core. And so obviously, Mini-Cactus and PGGB are natural candidates for being modules that will be totally suited. So if at some point you guys want to explore NF-Core and all of this aspect, this is really something we'd be very happy to discuss with you.
Yes, thank you. I'm quite aware of PGGB on NF-Core. And if you remember at the point where I was showing the path to generating the pangenome, I had to ditch the natural pathway of using PGGB because of the fact that the naming was not amenable to what it uses. And because of that, I mean, I wouldn't know the possibility of being able to manually intervene at that point using the NF-Core pipeline, because the pipeline, you put in your data, it runs to completion, but then at the end of it, I need to intervene to manually generate those VCF files. So I wouldn't know if it's going to be amenable to that.
So the NF-Core Slack is typically the kind of place where people help resolving this kind of questions. And so that's why I have advertised the Slack a little bit, because there is typically one channel per pipeline, and you'd be really, really welcome contributing to these things. And so without further ado, I let Francesca take over and give other people.
The next question is by Alexandru Mizeransky. Alexandru, do you want to unmute yourself or not? Okay.
Sure, thank you very much. Hi, thank you for the talk, it was very interesting. I was wondering, could you share some info about what sort of hardware infrastructure you used for the Pangenome graph construction? So CPUs, memory, storage, that sort of thing, and how long it took to build the two graphs, the PGGB and the Minigraph-Cactus one.
Okay, thank you for that great question. So for PGGB graph, in terms of building the pangenome, we're using CPU of up to 32 CPUs, and then memory footprint, like total memory footprint for constructing could be as high as 104 gigabytes. And then talking about runtime. So runtime for constructing the pangenome, it was done chromosome by chromosome. That was the best way, or the most tenable way of doing that. So depending on the length of the chromosome, because of the fact that we have some 3 very huge chromosomes in sheep, and then some others that are smaller. So for the very big chromosomes, we had up to 12 hours of wall time to build the pangenome. Then for the shorter ones, maybe like 8 hours of wall time using those compute resources.
Okay, thank you so much. The next question is by Lucia Giovannini from UniPG. Lucia, do you want to unmute yourself? If not, I will read it. So she is asking, do you suggest using both approaches of PGGB and MC, despite the fact that PGGB seems to capture a greater number of variants?
Yes, thank you for the question. I would say that your utility of the pangenome would actually determine which approach would be best for you. So for instance, if you're interested in using the pangenome, using the pipeline to call variants from either long reads or short reads, genotyping individuals, etc., then you want to use one that is amenable to easy indexing and alignment. And in that case, that would be a Minigraph-Cactus pangenome. because of the lesser complexity, you're able to easily use it to carry out the downstream pipeline of alignment and genotyping. But if what you're interested in is to really capture as many variation as possible that you have in your population, and you're not interested in perhaps the pipeline of alignment of reads and also genotyping, then you are perhaps just interested in looking at the structural variance between the different individuals that went to the pangenome, then you are better off using the PGGB approach, where you could just characterize the SVs and then maybe do a direct comparative analysis of the individuals that you have in your pangenome. So that's where using a PGGB approach would be very, very useful. So what you want to do with your pangenome, I would say, will determine which of the two tools you would use.
Okay, so we still have a couple of questions, and I think we have time a little bit, really short. So Golly is asking, when checking the known variants in the graph, does PGGB or Cactus can present better results?
So far, based on what we have seen, PGGB has been able to capture better the variants that we have in the pangenome. And just like I had mentioned earlier too, aside the fact that we're just checking the tool for concordance, we are more interested at this point in characterizing the SVs and all that. So we have more focus on the PGGB graph just because of the computational challenge of using what's it called, the pipeline for genotyping individuals by aligning reads and then going down with the VG-giraffe pipeline to do that. So I would say that so far, so good. We have more representation or better representation using PGGB.
Okay, so Shamim, do you want to unmute yourself if you are still here? Because you asked two questions. Shamim? If you're talking, we cannot hear you because I see that you're unmuted, but...
We can just read it if that's going to be quicker.
Yeah.
Okay.
So he's asking, I'm sorry, I will read it. He's asking, why do we get 26 haplotypes instead of 23 when including subspecies? And the second question is, how can we use pangenome in genomic prediction? Does it also follow a reference genome structure?
Okay, thank you so much for the question. So I had mentioned that we had 23 domestic sheep haplotypes and then 3 subspecies. So those two sets would make up the 26 haplotypes that I mentioned. Then for the second question, how we can use pangenome in genomic prediction. Of course, the use for pangenome, I mean, it's quite evolving. It's a very dynamic area. So many people are using it for different things. And like I'd mentioned earlier, you could use it for genotyping. For genomic prediction, I really don't.
You want me to answer that one?
Yeah, please go ahead. Yeah, thank you.
So I think for the genomic prediction, you might want to consider which breeds. So maybe you don't want to use a multi-species pangenome, but in a particular, if you do a pangenome within a particular species, if there's regions of the genome that are being associated with traits that are being passed on, then understanding where those are and how frequent they happen, and more specifically how they are correlate, how they are predictive of the phenotype that you're looking. That's where this kind of information can be informative, right? So if you just do SNPs and we just have a small number of SNPs and we use those for genomically enhanced EBVs, for example, right, the more information and the more precise information, the more accurate your predictions are actually going to be. That is the molecular genetics answer to that. Quantitative person might have a slightly different perspective.
Okay, so we have still one question on the chat, and then a question from Hubert Pausch. So Sylvia is asking, do the software you mentioned accept only short reads to build a pangenome? And what are the disadvantages of having only short reads?
So the pangenomes are not built of only short reads. The genomes that go into the pangenomes are a combination of short read, long read, but mostly long read. And your pangenome and your output from your pangenome is really subject to the quality of the genomes that go in there. So the short read piece came into, now I have this pangenome, can I use short read sequence data to call variants and then use the data from there? So I think there may have been a little bit confusion on that front. So we really want the pangenome, the genomes that go into the pangenomes is high quality. And that usually means that we need long reads in those assemblies.
Okay. Hubert, please unmute yourself and ask the question.
Yeah, thank you for an interesting talk. The question I have, I don't have an answer to that, and I'm curious to hear your thoughts on that. When we promised to use pangenomes, we promised them to use them because we wanted to unify the references in a way and want to avoid the situations where everyone is using their most favorite reference sequence. And now it seems we're going to build multiple pangenomes with different software for different species. Some call it super pangenomes, where you also include outgroup species. And I'm asking myself, what are your thoughts? Are we back to square one? Are we now not having multiple reference sequences, but multiple pangenomes? And what are your thoughts on that? Thank you.
Thanks, Hubert. I think with the cattle pangenome, I think we end up at the what Temitayo said is, what is the purpose? What do we want to use this tool for? I think when we first ventured into some of these pangenomes and building these pangenomes, maybe we underestimated how difficult it would be to use on a, I'll go back to that microscopic scale and macroscopic scale, right? So, I mean, we can build one big, huge pangenome for each species, but then do we create something that's so complex and has so much variation that in the end, we can't really use it. So I think, we have to think about what do we want to use this tool for? And I don't know that we really have an answer either. I don't know that the human pangenome consortium really has an answer. And it might be that we need a couple different tools to answer those questions, and a couple of different population resources to answer questions that are more specific for those, right? So if we really care about, I'm just going to pick a species, about Holstein dairy cattle, right? Do we include a bunch of other subspecies like gyr and a bunch of other things in there? If we're trying to answer the question, what does the gyr do and how can that contribute, and what does that genome have that can help us think more forward on that Holstein question, then maybe we do. But again, if we build this monster of a resource that in the end we can't use, or that becomes so complex to use, then it makes it rather difficult. So to answer your question, are we back where we started? I don't think we are back where we started, but I think that we need to think about the end use of the tools that we're building, which is a little bit why we took this sort of two breed approach. Not everybody cares about the mouflon and the bighorn. So if we just care about Ovis aries, then maybe that's the pangenome that we should use. And maybe if we care about comparative genomics, we should not have 23 domestic sheep in there, but maybe have a couple different domestic sheep and the other subspecies for a more comparative. So maybe the answer isn't one tool for everything or one resource for everything, but a couple different resources depending on the question that we're asking. That's kind of the approach that we've been taking a little bit. I don't know if that is going to be a solution for all of the different species, but I don't know. Interested to hear your thoughts as well, Hubert.
Thank you, Brenda. Thank you, Hubert. I think we're gonna have to stop this very interesting discussion here, unless you have a very short point you want to make, Hubert.
No, not really. I also don't have an answer to that, and I think there are different reasons to build different pangenomes, so we need to explore. Thank you.
To me, it reminds me what happened many years ago when we were building domain collections, because eventually, at the end of the day, a domain is the pangenome of a protein domain. And there were a lot of discussion as to introduce all the sequences, carefully selected sequences, and the sweet spot was always having as much data as possible, being as high quality or informative as possible. And I'm tempted to think that there is nothing in genomics that is not sensitive to the quantity of data you put in. So I have the impression that the most integrative methods help a lot. And also, we still have not looked at what AI can do to